Inside you right now are billions upon billions of microscopic machines, and they are working nonstop. They build, they transport, they send messages. They are literally the engines of life. Today, we're going to unfold the secrets of these incredible little machines, proteins. So let's go on a journey from a simple genetic blueprint all the way to a complex, functional megastructure.
So let's kick things off with a question to bring this right home. What do your hair, the skin on your face, and the blood running through your veins all have in common? Well, the answer is they're all made of proteins, and each and every one of them is perfectly shaped for its unique job.
Okay, let's dive right in. Proteins are, without a doubt, the most versatile molecules in our bodies. You really can think of them as life's ultimate multitaskers. They're not just passive building blocks. Oh no, they are active participants in pretty much every single process that keeps you alive and kicking.
And their resume is, frankly, unbelievable. They work as enzymes, speeding up chemical reactions by millions of times. They act as hormones, carrying critical signals all over the body. They provide structure, transport vital molecules, they let our muscles contract, and they even fight off diseases. So you gotta ask, how can one type of molecule do so many different things? The secret, the absolute key to all of this, is its unique, three-dimensional shape.
So to understand that shape, we've got to go back to the beginning. See, every great story is written with an alphabet, right? Well, the story of every single protein is written with the amino acid alphabet. And get this, there are just 20 common letters, or amino acids, but you can arrange them in a nearly infinite number of ways to write every protein story imaginable.
And what's just incredible is that every single one of those 20 amino acids shares this exact same core structure you see here. You've got a central carbon atom, an amino group on one side, and a carboxyl group on the other. But it's this one part, this R group, that changes. And that, my friends, makes all the difference.
So that R group, that's the wild card. It's what makes each amino acid totally unique. Some are big, some are tiny, some are positively charged, some are negative, and some don't have a charge at all. It is the specific chemical personality of this one little side chain that will ultimately dictate how the entire protein folds into its final, complex, and functional 3D shape.
Okay, so we have our 20 letters. Now what? How do we actually link them together to start writing the protein story? How do we turn these individual amino acids into a long chain? Kind of like forming a sentence.
Well, it all happens through this really simple and, honestly, super elegant process called a condensation reaction. Two amino acids line up next to each other, a water molecule gets kicked out, and then, snap, a strong covalent bond called a peptide bond forms, linking them together. This process just repeats over and over and over again, creating a long chain that we call a polypeptide.
And this diagram just shows it perfectly. You can see the hydroxyl group from one amino acid and a hydrogen from the other are pulled away. That's what forms the water. And then a super strong peptide bond forms right there between the carbon and the nitrogen. It's a beautifully efficient way to build.
Now, this specific, unique sequence of amino acids, all lined up in an order that's spelled out by our DNA, well, that has a name. We call it the protein's primary structure. Think of it as the most fundamental level of protein architecture. So the primary structure is basically a long, flexible string of beads. But here's the thing, and this is super important. At this stage, it's just a sequence. It can't actually do anything yet. It's useless. The real magic, the transformation into a working machine, that all happens in the folding.
All right, now this is where it gets really, really cool. That simple floppy chain now has to go through this incredibly intricate process of twisting and coiling and folding to get to its final functional 3D shape. This is the moment the chain actually becomes the machine.
The very first stage of folding creates what we call the secondary structure. And get this, it isn't even driven by those special R groups yet. Instead, weak little hydrogen bonds start to form between the atoms in the polypeptide's backbone. That repeating sequence of nitrogen, carbon, and carbon that forms the chain itself. These hydrogen bonds cause parts of the chain to do one of two things. It can either coil up into this beautiful spring-like shape on the left, called an alpha helix, or it can fold back and forth on itself, kind of like an accordion, to make what's called a beta-pleated sheet, like you see on the right. A lot of proteins actually have a mix of both.
But that's just the local folding. The main event, the real work of art, is the tertiary structure. Now, finally, those all-important R groups get to come out and play.
They start interacting with each other, pulling and twisting those helices and sheets into a very specific, compact, and final three-dimensional shape.
And to lock this final shape in place, the protein uses a whole toolkit of different bonds between the R groups. You've got more hydrogen bonds, you have stronger ionic bonds that form between charged groups, and even these super strong covalent bonds called disulfide bridges. And then there are hydrophobic interactions, which basically means the nonpolar R groups literally run and hide from the water around them, tucking themselves deep inside the protein's core.
So this whole complex folding process results in two major classes of proteins. You can almost think of them as two different architectural philosophies. So let's meet two titans from these opposing schools of design, the compact globular proteins and the long, strong, fibrous proteins.
This table breaks it down perfectly. You can see globular proteins are kind of like little spheres. They're compact and they dissolve in water. This makes them perfect for doing metabolic jobs inside our cells, like enzymes. Fibrous proteins, totally opposite. They're long, insoluble strands that are built for pure structural strength. So let's look at a superstar example of each.
First up, our globular superstar, hemoglobin. This is the protein inside your red blood cells, and it's responsible for one of the most critical jobs in your entire body, transporting oxygen. It is a true marvel of molecular engineering. Now, hemoglobin is extra special because it has what we call a quaternary structure. So what does that mean? It just means it's not one polypeptide chain. It's a team of 4 chains, 2 alpha and 2 beta chains, all working together. And each one holds a special non-protein part called a heme group. Right at the heart of each heme group is an iron ion, and that is what actually grabs onto an oxygen molecule. And here is where its shape is just pure genius. When you're in the lungs, where carbon dioxide levels are low, the hemoglobin molecule twists into a shape that has a really high affinity, a strong attraction for oxygen. It gets super sticky for oxygen and just greedily grabs onto it, loading up for its journey through the body. But then, as it travels out to your hard-working tissues like your muscles, the situation completely flips. Your cells are burning fuel, making lots of carbon dioxide. This makes the blood a little more acidic, and that forces the hemoglobin to change its shape again. And in this new shape, its affinity for oxygen absolutely plummets. It's not sticky anymore, and so it lets go, releasing its precious cargo right where it's needed most. It's just brilliant. Now let's pivot to our fibrous titan, collagen. If hemoglobin is the delivery truck, collagen is the steel scaffolding that literally holds your body together. It's actually the single most abundant protein in the animal kingdom.
Collagen gets its incredible strength from its elegant structure. It's made of 3 long polypeptide chains, and each one is a helix itself. Then, those 3 are woven together into a super strong triple helix, just like a biological rope. The structure, which is held together by countless hydrogen bonds, gives it immense tensile strength, making it the perfect material for our skin, tendons, ligaments, and bones.
So if there's one thing to take away from all of this, is that for proteins, structure dictates function. It is everything. From hemoglobin's clever shape-shifting to collagen's rope-like strength, its form defines what it can do. Which leaves us with a really big, really important question. If the architecture is everything, what happens when a protein misfolds, even just a little bit? That's a question with some pretty profound consequences for our health.